Molecular genetics, genome organization. The genome is defined as the totality of all the DNA of an organism. Most genomes, all eukaryotic and prokaryotic, and some viruses are made of DNA, but a few viruses have RNA genome. The eukaryotic genome consists of two distinct parts, nuclear genome and organelles, mitochondrial and chloroplast genome. The amount of DNA present in haploid genome of species is called C value. C value is characteristic of each species. The value ranges from less than 10 raised to 6 base pairs as in smallest prokaryote Mycoplasma to more than 10 raised to 11 base pairs for amphibians. The genomes of higher organisms contain a large excess of DNA. Flowering plants, birds, mammals, reptiles, amphibians, bony fishes, cartilaginous fishes, echinoderms, crustaceans, insects, mollusks, worms, fungi, algae, bacteria, mycoplasms, and viruses. So this diagram depicts the DNA content of the haploid genome of a range of phyla. The range of values within a phylum is indicated by the shaded area. And this is, flowering plants. Then comes amphibians, later bony fishes. Along with that, insects are there. Then comes approximately the mollusks. Along with that, bacteria is also there. Fungi. And the lowest amount is, most probably from mammals, birds, and crustaceans. Along with that, algae, like that. So, DNA content of the haploid genome is related to the morphological complexity of lower eukaryotes. DNA content, DNA content of the haploid genome is related to the DNA content of the haploid genome is related to the morphological complexity of lower eukaryotes, but varies extensively among the higher eukaryotes. In lower eukaryotic organisms like yeast, amount of DNA increases with increasing complexity of organisms. However, in higher eukaryotes, there is no correlation between increased genome size and complexity. This lack of correlation between genome size and genetic complexity is referred to as C-value paradox. For example, man is more complex than amphibians in terms of genetic development, but some amphibian cells contain 30 times more DNA than human cells. Moreover, the genomes of different species of amphibians can vary 100-fold in their DNA content. Okay, here we could see the genome sizes of different organisms like Saccharomyces cerevisiae, yeast, genome size MB, 12 MB, then mustard plant 120 MB, then fruit fly 170 MB, Homo sapiens 3300 MB, barley 5300 MB. Genome complexity. Genome complexity is the total length of different sequences of DNA. Genome complexity can be measured through the renaturation kinetics of denatured DNA. Genome complexity is the total length of different sequences of DNA. Genome complexity can be measured through the renaturation kinetics of denatured DNA. Renaturation of DNA occurs through the complementary base pairing. Renaturation of DNA depends on random collision of the complementary strands and follows second order kinetics. A DNA reassociation reaction is described by the quote half. If large DNA is sheared into uniform fragments and allowed to renature, then the rate of renaturation of denatured DNA is expressed as dC by dt is equal to minus kc square, where k is the second order rate constant, c is the concentration of single stranded DNA at a time t, and the second order rate equation for two complementary strands coming together is given by the rate of decrease in c. And starting with a concentration C0 of completely denatured DNA at t is equal to 0, the amount of single stranded DNA remaining at some time t is c by c0 is equal to 1 by 1 plus k c0t. The time of half of the DNA to renature when c by c0 is equal to 0.5 is defined as t is equal to t half, then 0.5 is equal to 1 by 1 plus k c0t half, and thus 1 plus k c0t half is equal to 2, yielding C0 t half is equal to 1 by k. The product of C0 into t half is called a coat half. It is inversely proportional to the rate constant, since the C0 T half is the product of the concentration and time required to proceed halfway. A greater coat half implies a slower reaction. The reassociation of DNA usually is followed in the form of a coat curve. A graph of the fraction of single-stranded DNA re-annealed is a function of C cot T in a semi-logarithmic plot is referred to as a coat curve. The rate of reassociation can be followed spectrophotometrically by the UV absorbance decrease as duplex DNA is formed. So, a coat curve describes the kinetics of reassociation of a sample of double stranded DNA that has been fragmented to small pieces, denatured to single strands, and then permitted to reanneal. The coated half of a reaction indicates the total length of different sequences that are present. This is described as the kinetic complexity, and the coat half of the renaturation of the DNA of any genome is proportional to its complexity. The complexity of any DNA can be determined by comparing its coat half with that of a standard DNA of known complexity. Repetitive DNA sequence is first identified by studies of rates of DNA reassociation. What relative rates of reassociation are expected for sequence repeated 100 times in the genome compared to genes with only a single copy, 1,000 times? DNA association is a bimolecular reaction, so the reaction rate is proportional to the square of the DNA concentration. A sequence repeated 1,000 times in the genome would therefore reassociate with a rate of 10 raised to 6-fold greater than that of a single copy DNA. Reassociation kinetics identifies two general types of genomic sequences. Non-repetitive DNA consists of a sequence that are unique. There is only one copy in a haploid genome, and a repetitive DNA, that means sequences that are present in more than one copy in a haploid genome. So repetitive sequences show more rapid renaturation rates than the non-repetitive DNA sequences. The proportion of the non-repetitive and repetitive DNA sequences varies widely. So prokaryotes contain only non-repetitive DNA. Similarly, in lower eukaryotes, most of the DNA is non-repetitive. In higher eukaryotes, up to half of the DNA often is occupied by repetitive components. In case of plants and amphibians, repetitive components may account for up to 80% of the genome. In general, the length of the repetitive DNA component tends to increase with overall genome size. Renaturation kinetics further divide the repetitive DNA sequences into two general classes, like moderately repetitive DNA sequences and highly repetitive DNA sequences. Highly repetitive sequences. Highly repetitive DNA sequences or leads consists of a very short sequences repeated typically less than 100 base pair many times in tandem in large clusters. Because of its short repeating unit, it is sometimes also known as a simple sequence DNA. Such simple sequence DNA can often be separated from the bulk of the chromosomal DNA by shear degradation followed by density gradient ultracentrifugation in CSCL. So equilibrium density gradient ultracentrifugation in CSCL constitute most common method for DNA separation. It separates the DNA on the basis of their differences in bind density. The bind density rho of double-stranded csDNA depend on its base composition. Rho is equal to 1.660 plus 0.098 x g plus c. Distinctive base composition of a simple sequence DNA and thus differences in bind density result in the formation of satellite to the main DNA band. The sequence of this DNA is known as satellite DNA. For example, when mouse DNA is centrifuged on a density gradient, two types of bands may be distinguished: main band and a satellite. The main band contains 92% of the genome and is centered on a bind density of 1.701 gram centimeter cube. The smaller peak represents 8% of genome and has a distinct bind density of 1.690 gram centimeter cube. So satellite DNAs are species specific. A single genome can contain several different types of satellite DNA, each with a different repeated unit. These units being anything from less than 5 to 200 base pair. Satellite DNA sequences generally are not transcribed and are located most often in the heterochromatin associated with the centromeric regions of chromosomes. So satellites are present in many eukaryotic genomes. They may be either heavier or lighter than the main band, One type of human satellite DNA, the alphoid DNA, repeats only in the centromere region of chromosome. Human genomic DNA contains a main band and three satellite bands are fractioned by density gradient centrifugation. Mini satellites and microsatellites. Mini satellites and microsatellites are randomly repeated DNA and also classified as satellite DNA, but not appearing in satellite bands or density gradients. So many satellites form clusters up to 20 kb in length with 20 to 50 repeat units, each containing 50 to about 100 base pairs. Telomeric DNA, which in human comprises hundreds of copies of motif in an example of mini satellite. Mini satellite clusters are shorter, usually 150 base pairs, and the repeat unit is usually 13 base pair or less. The typical microsatellite consists of a 1, 2, 3, or 4 BP unit repeated 1 at 10 to 20 times. And satellite DNA mainly at centromere, alpha satellite, centromeric heterochromatin, beta satellite, centromeric heterochromatin of some chromosomes, and mini satellite DNA at or closed telomere, microsatellite DNA dispersed throughout all chromosomes. Then DNA fingerprinting, also called DNA testing, DNA typing, DNA profiling, is a technique used to distinguish between individuals of the same species, usually only samples of their DNA using. Okay, so the chemical structure of everyone's DNA is the same. The only difference between persons is the order of the base pairs. Hence, every person could be identified solely by the sequence of their base pairs. Although two individuals will have the vast majority of their DNA sequences in common, DNA profiling exploits highly variable repeat sequences called VNTRs, variable number tandem repeat. VNTR is a general term which includes microsatellite VNTR and mini satellite VNTR, but is often used loosely to mean the mini satellite class. Within a species, the nucleotide sequence of the repeat units composing VNTR are highly conserved among individuals. However, differences in the number of repeats and thus in the lengths of simple sequence tandem arrays containing the same repeat unit are quite common among individuals. These DNA polymorphisms often form the basis of DNA fingerprinting. Variable number tandem repeat. The high variability in mini satellites makes them especially useful for genome mapping because there is a high probability that individuals will vary in their alleles at such a locus. In human chromosomes, many VNTR regions are broadened by restriction to nucleus sites on either side. Alleles of a given VNTR differ from each other in the size of the segment by the restriction in the nucleus, giving rise to nearly unique RFLP patterns. This is the basis of the technique known as DNA fingerprinting. With this technique, forensic scientists have been able to find missing persons, determine whether or not an individual is the parent of a child, and establish beyond reasonable doubt that a person is guilty of a crime for what he or she has been accused. Then moderately repetitive sequences. Moderately repetitive DNA consists of relatively short sequences that are repeated typically 1 to 10,000 in the genome. These sequences are dispersed throughout the genome. A significant fraction of the moderately repetitive DNA consists of mobile DNA elements. Okay, so moderately repetitive DNA consists of relatively short sequences that are repeated typically like this rate in the genome. The sequences are dispersed throughout the genome. A significant fraction of the moderately repetitive DNA consists of mobile DNA elements or transposable elements, short sequences of DNA that have the ability to move to new locations in the genome and or to make additional copies of themselves. However, most of these elements move only very rarely. Transposons are considered as selfish DNA, which is defined as sequences that do not contribute to genotype and organisms but propagate within genome. Then mobile DNA elements. Mobile DNA elements are the moderately repeated DNA sequences interspaced at multiple sites throughout the genome. Genomes generally contain many varieties of transposable elements. Mobile DNA elements encode enzymes that can insert their sequence into new sites in genomic DNA. The process by which these sequences are copied and inserted into new site in the genome is called a transposition. Mobile DNA elements are of two types, like transposons and retrotransposons. And replicator transposition is also there. Conservative and replicator. Thank you for listening. Tomorrow we'll be learning some important aspects related to other bacterial mobile elements.